Mineral nutrition. The basic needs of all living organisms are essentially the same. They require macromolecules such as carbohydrates, proteins, and fats, and water and minerals for their growth and development. This chapter focuses mainly on inorganic plant nutrition, wherein you will study the methods to identify elements essential to growth and development of plants and of the criteria for establishing the essentiality. You will also study the role of the essential elements, their major deficiency symptoms, and the mechanism of absorption of these elements. The chapter also introduces you briefly to the significance and the mechanism of biological nitrogen fixation. 12.1 Methods to study the mineral requirements of plant. In 1860, Julius von Sachs, a prominent German botanist, demonstrated for the first time that plant could be grown to maturity in a defined nutrient solution in complete absence of soil. This technique of growing plants in a nutrient solution is known as hydroponics. Since then, a number of improvised methods have been employed to try and determine the mineral nutrition essential for plants. The essence of all these methods involves the culture of plants in a soil-free, defined mineral solution. These methods require 12.1 Methods to study the mineral requirements of a plant. In 1860, Julian von Sachs, a prominent German botanist, demonstrated for the first time that plants could be grown to maturity in a defined nutrient solution in complete absence of soil. This technique of growing plants in a nutrient solution is known as hydroponics. Since then, a number of improvised methods have been employed to try and determine the mineral nutrients essential for plants. The essence of all these methods involves the culture of plants in a soil-free, defined mineral solution. These methods require purified water and mineral nutrient salt. Can you explain why is this so essential? After a series of experiments in which the roots of the plants were immersed in nutrient solutions and wherein an element was added or substituted or removed or given in varied concentration, a mineral solution suitable for the plant growth was obtained. By this method, essential elements were identified and their deficiency symptoms discovered. Hydroponics has been successfully employed as a technique for the commercial production of vegetables such as tomato, seedless cucumber of lettuce, and lettuce. It must be emphasized that the nutrient solutions must be adequately aerated to obtain the optimum growth. What would happen if solutions were poorly aerated? Diagrammatic views of the hydroponic technique is given in figure 12.1 and 12.2. Essential mineral nutrients. Most of the minerals present in the soil can enter plants through roots. In fact, more than 60 elements of the 105 discovered so far are found in the different plants. Some plant species accumulate selenium, some others gold, while some plants growing near nuclear test sites takes up radioactive strontium. There are techniques that are able to detect the minerals even at a very low concentration, 10 to the power minus 8 gram per milliliter. The question is whether all the diverse mineral elements present in a plant, for example gold and selenium as mentioned above, are really necessary for plants. How do we decide what is essential for plants and what is not? 12.2.1 Criteria for Essentiality. The criteria for essentiality of an element are given below. A. The element must be absolutely necessary for supporting normal growth and reproduction. In the absence of the element, the plant do not complete their life cycle or set the seeds. B. The requirement of the element must be specific and not replaceable by another element. In other words, deficiency of any one element cannot be met by supplying some other element. C. The element must be directly involved in the metabolism of plants. Based on based above Based upon the above criteria, only a few elements have been found to be absolutely essential for plant growth and metabolism. These elements are further divided into two broad categories based on their quantitative requirements. First is macronutrients and second is micronutrients. Macronutrients are generally present in plant tissues in large amounts in excess of 10 mole per kilogram of dry matter. The macronutrients include carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium, and magnesium. Of these, carbon, hydrogen, oxygen are mainly obtained from carbon dioxide and water, while the others are absorbed from the soil as mineral nutrition. Micronutrients or trace elements are needed in very small amounts, less than 10 mole per kilogram of dry matter. These include iron, manganese, copper, molybdenum, zinc, boron, chlorine, and nickel. In addition to the 17 essential elements named above, there are some beneficial elements such as sodium, silicon, cobalt, and selenium. They are required by higher plants. Essential elements can also be grouped into four broad categories on the basis of their diverse functions. These categories are essential elements as components of biomolecules and hence structural elements of cells. Example: carbon, hydrogen, oxygen, and nitrogen. Essential elements that are components of energy related chemical compounds in plants. Example: magnesium in chlorophyll and phosphorus in ATP. Essential elements that activate or inhibit enzymes. For example, magnesium 2 positive is an activator of both ribose biphosphate carboxylase oxygenase and phosphoenol pyruvate carboxylase, both of which are critical enzymes in photosynthetic carbon fixation. Zn2 positive is an activator of alcohol dehydrogenase and molybdenum of nitrogenase during nitrogen metabolism. Can you name a few more elements that fall in this category? For this, you will need to recollect some of the biochemical pathways you have studied earlier. Some essential elements can alter the osmotic potential of the cell. Potassium plays an important role in the opening and closing of stomata. You may recall the role of minerals as solute in determining the water potential of a cell. 12.2.2 Role of macro and micronutrients. Essential elements perform several functions. They participate in various metabolic processes in the plant cells such as permeability of cell membrane, maintenance of osmotic concentration of cell sap, electron transport system, buffering action, enzymatic activity, and act as major constituents of macromolecules and coenzymes. Various forms and functions of essential nutrients elements are given below. Nitrogen, this is the essential nutrient element required by plants in the greatest amount. It is absorbed mainly as NO3 negative, though some are also taken up as NO2 negative or NH4 positive. Nitrogen is required by all parts of a plant, particularly the meristematic tissues and the metabolically active cells. Nitrogen is one of the major constituents of proteins, nucleic acids, vitamins, and hormones. Phosphorus is absorbed by the plants from soil in the form of phosphate ions, either as H2PO4 positive or HPO4 2 positive. Phosphorus is Phosphorus is absorbed by the plants from soil in the form of phosphate ions, either as H2PO4 negative or HPO4 2 positive. Phosphorus is a constituent of cell membranes, certain proteins, all nucleic acids, and nucleotides, and is required for all phosphorylation reactions. Potassium, it is absorbed as potassium ion K positive in plants, that is required in more abundant quantities in the meristematic tissues, buds, leaves, and root tips. Potassium helps to maintain an anion-cation balance in the cells and is involved in protein synthesis, opening and closing of stomata, activation of enzymes, and the maintenance of turgidity of cells. Calcium. Plants absorb calcium from the soil in the form of calcium ions Ca2 positive. Calcium is required by meristematic and differentiating tissues. During cell division, it is used as the synthesis of cell wall, particularly as calcium pectate in the middle lamella. It is also needed during the formation of mitotic spindle. It accumulates in older leaves. It is involved in the normal functioning of cell membrane. It activates certain enzymes and plays an important role in regulating metabolic activities. Magnesium, it is absorbed by plants in the form of divalent Mg2 positive. It activates the enzymes of respiration, photosynthesis, and are involved in the synthesis of DNA and RNA. Magnesium is a constituent of the ring structure of chlorophyll and helps to maintain the ribosome structure. Sulfur. Plants obtain sulfur in the form of sulfate SO4 2 negative. Sulfur is present in two amino acids, cytosine and methionine, and is the main constituent of several coenzymes, vitamins, thiamine, biotin, coenzyme A, and ferredoxin. Iron. Plants obtain iron in the form of ferric ions Fe3 positive. It is required in larger amount in comparison to other micronutrients. It is an important constituent of proteins involved in the transfer of electrons like ferredoxin and cytochromes. It is reversibly oxidized from Fe2 positive to Fe3 positive during electron transfer. It activates catalase enzyme and is essential for the formation of chlorophyll. Manganese, it is absorbed in the form of magnesium Manganese ions Mn2 positive, it activates many enzymes involved in photosynthesis, respiration, and nitrogen metabolism. The best defined function of manganese is the splitting of water to liberate oxygen during photosynthesis. Zinc, plants obtain zinc as it into positive ions. It activates various enzymes, especially carboxylases. It is also needed in the synthesis of oxygen. Copper, it is absorbed as cupric ion CO2 positive. It is essential for the overall metabolism in plants. Like iron, it is associated with certain enzymes involved in redox reactions and is reversibly oxidized from Co positive to CO2 positive. Boron, it is absorbed as BO3 3 negative or B4O7 2 negative. Boron is required for uptake and utilization of CO2 positive, membrane functioning, pollen germination, cell elongation, cell differentiation, and carbohydrate translocation. Molybdenum, plants obtain it in the form of molybdate ions, MOO2 positive. It is a component of several enzymes, including nitrogenase and nitrate reductase, both of which participate in nitrogen and metabolism. Chlorine, it is absorbed in the form of chloride anion Cl negative along with Na positive and K positive. It helps in determining the solute concentration and the anion cation balance in cells. It is essential for the water splitting reaction in photosynthesis, a reaction that leads to oxygen evolution. 12.2.3 Deficiency symptoms of essential elements. Whenever the supply of an essential element becomes limited, the plant growth is retarded. The concentration of the essential element below which plant growth is retarded is termed as a critical concentration. The element is said to be deficient when present below the critical concentration. Since each element has one or more specific structural or functional role in plants, in the absence of any particular element, plants show certain morphological changes. These morphological changes are indicative of certain element deficiencies and are called deficiency symptoms. The deficiency symptoms vary from element to element and they disappear when the deficient mineral nutrient is provided to the plant. However, if deprivation continues, it may eventually lead to the death of the plant. The parts of the plant that show the deficiency symptoms also depend on the mobility of the elements in the plant. For elements that are actively mobilized within the plants and exported to young developing tissues, the deficiency symptoms tend to appear first in the older tissues. For example, the deficiency symptoms of nitrogen, potassium, and magnesium are visible first in the senescent leaves. In the older leaves, biomolecules containing those elements are broken down, making those elements available for mobilizing to younger leaves. The deficiency symptoms tend to appear first in the young tissues whenever the elements are relatively immobile and are not supported out of the mature organs. For example, elements like sulfur and calcium are a part of the structural component of the cell and hence are not easily released. The aspect of mineral nutrient plants is of great significance and importance to agriculture and horticulture. The kind of deficiency symptoms shown in the plants include chlorosis, necrosis, stunted plant growth, premature fall of leaves and buds, and inhibition of cell division. Chlorosis is the loss of chlorophyll leading to yellowing in leaves. This symptom is caused by the deficiency of elements nitrogen, potassium, magnesium, sulfur, iron, manganese, zinc, and molybdenum. Likewise, necrosis or death of particularly leaf tissue, is due to the deficiency of calcium, magnesium, copper, potassium, or lack of low level of nitrogen, potassium, sulfur, and molybdenum causes an inhibition of cell division. Some elements like nitrogen, sulfur, molybdenum delay flowering if their concentration in plant is low. You can see from the above that deficiency of any element can cause multiple symptoms and that the same symptoms may be caused by the deficiency of one or several different elements. Hence, to identify the deficient elements, one has to study all the symptoms developed in all the various parts of the plant and compare them with the available standard tables. You must also be aware that different tables also respond differentially to the deficiency of the same element. I'm sorry, I am reading this line once again. We must also be aware that different plants also respond differently to the deficiency of the same element. 12.2.4 Toxicity of micronutrients. The requirement of micronutrients is always in low amounts, while their moderate decrease causes the deficiency symptoms and the moderate increase causes toxicity. In other words, there is a narrow range of concentration at which the elements are optimum. Any mineral iron concentration in tissues that reduces the dry weight of tissues by about 10% is considered toxic. Such critical concentration vary widely among different micronutrients. The toxicity symptoms are difficult to identify. Toxicity levels for any element also vary for different plants. Many a times, excess of an element may inhibit the uptake of another element. For example, the prominent symptom of manganese toxicity is the appearance of brown spots surrounded by chlorinic veins. It is important to note that manganese competes with iron and manganese for uptake and with magnesium for binding with enzymes. Manganese also inhibits calcium translocation in shoot apex. Therefore, excess of manganese may in fact induce deficiencies of iron, magnesium, and calcium. Thus, what appears as symptoms of manganese toxicity may actually be the deficiency symptoms of iron, manganese, and calcium. Can this knowledge be of some importance to a farmer, a gardener, or even for you in your kitchen garden? 12.3 Mechanism of absorption of elements. Much of the studies on mechanism of absorption of elements by plants has been carried out in isolated cells, tissues, or organs. These studies reveal that the process of absorption can be demarcated into two main phases. It is the first phase, an initial rapid uptake of ions into the free space or outer space of cells. The apoplast is passive. In the second phase of uptake, the ions are taken in slowly into the inner space, the symplast of the cells. The passive movement of ions into the apoplast usually occurs through ion channels, the transmembrane proteins that functions as selective pores. On the other hand, the entry or exit of ions to and from the symplast requires the expenditure of metabolic energy, which is an active process. The movement of ions is usually called flux. The inward movement into the cells is influx and the outward movement efflux. You have read the aspect of mineral nutrient uptake and translocation in plants in chapter 11. 12.4 Translocation of solutes. Mineral salts are translocated through xylem along with the ascending mineral of water, which is pulled up through the plant by transpirational pull. Analysis of xylem sap shows that the presence of mineral salts in it that the mineral salts are present in it. Use of isotopes of mineral elements also substantiate the view that they are transported through the xylem. You have already disclosed or discussed the movement of water in xylem in chapter 11. 12.5 Soil is reservoir of essential elements. Majority of the nutrients that are essential for the growth and development of plants become available to the roots due to weathering and breaking down or breakdown of rocks. These processes enrich the soil with dissolved ions and inorganic salts. Since they are derived from the rock minerals, their role in plant nutrition is referred to as mineral nutrition. Soil consists of a wide variety of substances. Soil not only supplies minerals but also harbors nitrogen-fixing bacteria, other microbes, holds water, supplies air to the roots, and acts as matrix that stabilizes the plant. Since deficiency of essential minerals affect the crop yield, there is often a need for supplying them in through fertilizers. But both macronutrients sodium, potassium, phosphorus, sulfur, etc., and micronutrients copper, zinc, iron, manganese, etc., form components of fertilizers and are applied as per need. 12.6 Metabolism of nitrogen. 12.6.1 Nitrogen cycle. Apart from carbon, hydrogen, and oxygen, nitrogen is the most prevalent element in living organisms. Nitrogen is a constituent of amino acids, proteins, hormones, chlorophylls, and many of the vitamins. Plants compete with microbes for the limited nitrogen that is available in soil. Thus, nitrogen is a limiting nutrient for both natural and agricultural ecosystems. Nitrogen exists as two nitrogen atoms joined by a very strong triple covalent bond that is given over here, N triple bond N. The process of conversion of nitrogen into 2 ammonia is termed as nitrogen fixation. In nature, lightning and ultraviolet radiation provide enough energy to convert nitrogen to nitrogen oxides, NO, NO2 or N2O. Industrial combustion, forest fires, automobile exhaust, and power generating stations are also sources of atmospheric nitrogen oxides. Decomposition of organic nitrogen of dead plants and animals into ammonia is called ammonification. Some of this ammonia volatilizes and re enters the atmosphere, but most of it is converted into nitrate by soil bacteria in the following steps. 2NH3 plus 3O2 gives 2NO2 plus 2H positive plus 2H2O. 2NO2 negative plus O positive gives, 2NO2 negative plus oxygen gives 2NO3 negative. Ammonia is first oxidized to nitrite by the bacteria Nitrosomonas and or Nitrococcus. The nitrite is further oxidized to nitrate with the help of the bacterium Nitrobacter. These steps are called nitrification. These nitrifying bacteria are chemotrophs. The nitrate thus formed is absorbed by plants and is transported to the leaves. In leaves, it is reduced to form ammonia that finally forms the amine group of amino acids. Nitrate present in the soil is also reduced to nitrogen by the process of denitrification. Denitrification is carried by bacteria Pseudomonas and Theobacillus. 12.6.2 Biological nitrogen fixation. Very few living organisms can utilize the nitrogen in the form of N2 or dinitrogen available abundantly in the air. Only certain prokaryotic species are capable of fixing nitrogen. Reduction of nitrogen to ammonia by living organism is called biological nitrogen fixation. The enzyme nitrogenase, which is capable of nitrogen reduction, is present exclusively in prokaryotes. Such microbes are called dinitrogen fixers or nitrogen fixers. Now, N triple bond N in the presence of nitrogenase forms NH3. The nitrogen fixing microbes could be free living or symbiotic. Examples of free living nitrogen fixing aerobic microbes are Azotobacter and Beijerichia, while Rhodospirillum is anaerobic and free living. In addition, a number of cyanobacteria such as Enabena and Nostoc are also free living nitrogen fixers. Symbiotic biological nitrogen fixation. Several types of symbiotic biological nitrogen fixation associations are known. The most prominent among them is the legume-bacteria relationship. Species of rod-shaped rhizobium have such relationship with roots of several legumes such as alfalfa, sweet clover, sweet pea, lentils, garden pea, broad bean, clover beans, etc. The most common association on roots is as nodules. These nodules are small outgrowths on the roots. The microbe Frankia also produces nitrogen-fixing nodules on the roots of non-leguminous plant. Examples: Ulnus. Both Rhizobium and Frankia are free living in soil, but as symbionts can fix atmospheric nitrogen. Uproot any one plant of the common pulse just before flowering. You will see near spherical outgrowth on the roots. These are nodules. If you cut through them, you will notice that the central portion is red or pink. What makes the nodules pink? This is due to the presence of leguminous hemoglobin or leg hemoglobin. Nodule formation involves a sequence of multiple interactions between rhizobium and root of the host plant. Principal stages in the nodule formation are summarized as follows. Rhizobia multiply and colonize the surrounding of roots and gets attached to epidermal and root hair cells. The root hairs curl and the bacteria invade the root hair. An infection thread is produced carrying the bacteria into the cortex of the root, where they initiate the nodule formation in the cortex of the root. Then the bacteria are released from the thread into the cells of the root cells, which lead to the differentiation of specialized nitrogen fixation cells. The nodule thus formed establishes a direct vascular connection with the host for exchange of nutrients. These events are depicted in figure 12.4. The nodule contains all necessary biochemical components such as the enzyme nitrogenase and leg hemoglobin. The enzyme nitrogenase is molybdenum iron protein and catalyzes the conversion of atmospheric nitrogen to ammonia, the first stable product of nitrogen fixation. Figure 12.4 is given over here. You can see it a bit later. The reaction is as follows. Nitrogen or dinitrogen plus 8 electrons plus 8 hydronium ions plus 16 ATP gives 2NH3 plus H2 plus 16 ADP plus 16 Pi. The enzyme nitrogenase is highly sensitive to the molecular oxygen. It requires anaerobic condition. The nodules have adaptations that ensures that the enzyme is protected from oxygen. To protect these enzymes, the nodule contain an oxygen scavenger called leg hemoglobin. It is interesting to note that these microbes live as aerobes under free living conditions where nitrogenase is not operational, but during nitrogen fixing events they become anaerobic, thus protecting the nitrogenase enzyme. You must have noticed in the above reaction that the ammonia synthesis by nitrogenase requires a very high input of energy, 8 ATP for each NH3 produced. The energy required thus is obtained from the respiration of the host cells. Fate of ammonia at physiological pH, the ammonia is protonated to form NH4 positive ammonium ions. While most of the plants can assimilate nitrate as well as ammonium ions, the latter is quite toxic to plants and hence cannot accumulate in them. Let us now see how the NH4 positive is used to synthesize amino acids in plants. There are two main ways in which this can take place. Reductive amination. In these processes, ammonia reacts with alpha ketoglutarate acid and forms glutamic acid as indicated in the equation given below. Alpha ketoglutarate acid plus NH4 positive plus NADPH gives glutamate or dehydrogenase. Glutamate plus water plus NADP. Transamination. It involves the transfer of amino groups from one amino acid to the keto group of a keto acid. Glutamic acid is the main amino acid from which the transfer of NH to the amino group takes place, and the other amino acids are formed through transamination. The enzyme transaminase catalyzes all such reactions, for example, amino donor, amino acceptor, and the other products given over there. The two most important amides, asparagine and glutamine, found in plants are a structural part of proteins. They are formed from two amino acids, namely aspartic acid and glutamic acid, respectively, by addition of another amino group to each. The hydroxyl part of the acid is replaced by another NH2 negative radical. Since amides contain more nitrogen than the amino acids, they are transported to other parts of the plant via xylem vessels. In addition, along with the transpiration stream, the nodules of some plants, example soybean, export the fixation nitrogen as ureates. These compounds also have a particularly high nitrogen to carbon ratio. Plants obtain their inorganic nutrients from air, water, and soil. Plants absorb a wide variety of mineral elements. Not all the mineral elements that they absorb are required by plants. Out of the more than 105 elements discovered so far, less than 21 are essential and beneficial for normal plant growth and development. The elements required in large quantities are called macronutrients, while those required in less quantities or in trace are termed as micronutrients. These elements are either essential constituents of proteins, carbohydrates, fats, nucleic acid, etc., and or take part in various metabolic processes. Deficiency of each of these essential elements may lead to symptoms called deficiency symptoms. Chlorosis, necrosis, stunted growth, impaired cell division are some prominent deficiency symptoms. Plants absorb mineral through roots by either passive or active processes. They are carried to all parts of the organism through xylem along with water transport. Nitrogen is very essential for the sustenance of life. Plants cannot use atmospheric nitrogen directly, but some of the plants in association with nitrogen fixing bacteria, especially roots of legumes, can fix this atmospheric nitrogen into biologically usable forms. Nitrogen fixation requires a strong reducing agent and energy in the form of ATP. Nitrogen fixation is accomplished with the help of nitrogen fixing microbes, mainly rhizobium. The enzyme nitrogenase, which plays an important role in biological nitrogen fixation, is very sensitive to oxygen. Most of the processes takes place in anaerobic environment. The energy ATP required is provided by the respiration of the host cells. Ammonia produced following nitrogen fixation is incorporated into amino acids as the amino group. Okay guys, thanks a lot for being consistent. I have stopped a lot in between, but I'll try not taking any kind of pauses from on the next time. Let's keep on motivating each other. Let's keep on staying consistent. Thank you, and we'll crack this thing. Goodbye.